Hello and welcome viewers. Today we are going to cover a very important topic of the CSIR NET Life Science syllabus, which is regarding cell signaling. This is the unit 4 in the syllabus. We will try to look at the components which are involved. For example, there would be a ligand, there would be some receptor, second messengers, and the downstream molecules which they are affecting. There are different types of signaling processes, signaling pathways. This video is beyond the scope of covering all of them, but we'll try to look at the most important ones. Now, whenever you think about signaling, it means one cell giving a signal to some other cell. It can be with a direct contact or with some diffusible molecules which one cell releases extracellularly. Those molecules which are called as ligands, they can come and bind to receptors which are mostly membrane bound towards the plasma membrane. But they can be also cytosolic and sometimes they can be also some nuclear receptors as well. The most significant type of receptor that we see is the GPCR or G protein coupled receptor. Apart from that, you can also have enzyme linked receptor in which there would be an enzymatic action linked with the receptor. Sometimes you will have some cell matrix adhesion receptors, and this part is a separate aspect of unit 4, which is cell communication with adhesion molecules. Sometimes there are ion channels and transporters also. This is also a way of cell signaling. And whenever we have this signaling, it affects the gene expression or some changes inside the cell. The changes can be physical, like making the cells shape change or making the cells move. That would be affecting the cytoskeleton, or some organelles for them to undergo apoptosis, or they want to undergo differentiation. Then the changes would be in the organelles, or else most of the time the changes that we will see is in the nucleus, that is in the DNA. So the molecules downstream of the molecule, there would be some transcription factors which can affect the expression, gene expression in the cell differently. This entire thing comes in the form of a cascade. Cascade means a sequence of events, and you are supposed to know all the elements which are present in this entire cascade. Let's dive into some more detail. Before moving any forward, let me remind you that this topic will be common in many entrance exams. So if you are watching this video and in case you are preparing for any of these exams, then this topic can be covered from this video. Okay, that is GATE, ICMR, TIFR, ICR, DBT, and in fact many other such exams. So first of all, going for the introduction, we know that cells must respond adequately to external stimuli to survive. Cells will respond to stimuli via cell signaling. Some signal molecules enter cells, others will bind to cell surface receptors. And cellular responses will depend on some specific combination of signals. No signal often results in activation of apoptosis. Basically, a cell can have signals from many sources which can give it a signal to survive. Also, there can be some combination of signals which will tell the cell to not only survive but also to divide. Along with survival signal, you may have the cell going into a different type of gene expression which will make the cell express a specific set of genes and thus differentiate into a certain cell type. And as I said, if there is no cell signaling, then that might be a signal for the cell to die. That means undergo apoptosis. What are the basic elements of cell signaling? As I said, there should be some extracellular signal molecule. From where is this coming? From any other cell, which will be binding to a receptor protein. This receptor protein is present on the plasma membrane of the target cell. So this receptor protein, once it gets activated after binding of the molecule, this is going to trigger or activate several other downstream proteins inside the cell. So these will also act as signaling proteins, like this will signal this one, this will signal this one. But these are secondary messengers. The original messenger was these, so these will serve as secondary messengers, and most importantly, these are intracellular. Now these proteins, they will activate some more proteins which are the effector proteins. These effector proteins, they may be metabolic enzyme, there can be some transcription regulatory protein, or it can be a cytoskeletal protein as well. If it is a cytoskeletal protein, then you will see the changes in the cell shape or movement. If it is a transcription regulatory protein, then we will see change in the gene expression. And if it is an enzyme, then we can see an altered metabolism in this process. So as you can understand, signaling pathways consist of a series of proteins, and they should be in the exact same order. Each protein in the pathway alters the conformation of the next protein. So until and unless one order is achieved, the next sequence of event cannot take place. How the activations and inhibitions are generally made? The most common one is protein conformational changes. Because as we know, for a protein to be functional, we need a certain conformation of the protein, which is the native fold, a 3D fold, which will be functional, isn't it? If there is a disturbance of this conformation by binding of some activator or inhibitor, then the protein may become non-functional. So this is usually altered by phosphorylation. We know kinases will phosphorylate proteins, and that might turn on or turn off a particular enzyme. So basically they act as switches. Target proteins ultimately receive a message to alter the cell activity. Ultimately these target ones will receive the message to alter. This overall process is what is called as signal transduction. Transduction means from some other source, and then through some sequence of events it is creating a change inside the cell. So it is transduced through the entire pathway. Now in this phenomenon, there are two major ways in which a signal can be transduced. A mechanical or chemical stimulus is converted into a specific cellular response. It starts with a signal to a receptor and it ends with a change in the cell function or response. Now when we have a signaling cell, so let's talk about this signaling pathway first. There is a receptor over here. Now when this receptor is bonded with the ligand, some other effector molecule gets activated by binding with the activated receptor. Now because of this effector activation, it makes a new type of molecule. It generates another messenger, okay? So it's not this original messenger, but in turn it generates another messenger, which is called as a second messenger. The second messenger will be then binding with some other molecule inside the cell, with leading to changes in further downstream molecules as well, and ultimately a target protein is activated. That target protein can be anything. Out of the functions which I told you, with what changes can be done by cell signaling, the target protein can be anything. And another way in which this transduction can be achieved is when the signal will bind with the receptor. So as you can see, the receptor is getting phosphorylated. Okay, so in these kind of receptors, these are called as RTKs or receptor tyrosine kinase. In the receptor only there is a domain which is called as a kinase. Now we know kinase phosphorylates molecules. Whenever the ligand will come and bind with the receptor, this molecule kinase domain is activated, and with that it will phosphorylate itself. Before binding to the ligand, this was not phosphorylated, hence these proteins were not able to bind. But now that it is phosphorylated, these other proteins will dock on this surface, just like, binding to a charging point. So something like that. So they will dock at this particular point because of the conformational change. They will be able to do so. And by that, again, a series of effector proteins, they will be activated, finally activating the target protein. Okay. So receptors on or in target cells receive the message. Some cell surface receptors generate an intracellular second messenger through an enzyme called as an effector. Other surface receptors recruit proteins to their intracellular domains. Right? So cell to cell communication is of two types. One is intercellular, another one is intracellular. Intra means within the cell, inter means between two cells. So both type of communication you can have. If it is intercellular, in most of the cases what it is, one can be contact dependent, another can be through the release of a secreted molecule, which goes to the other cell where receptor will be present. So contact dependent means it is called as juxtacrine receptor. One example of this pathway is notch and delta signaling. Okay, one example of this kind of signaling pathway. Another one is secreted molecule. In secreted molecule, you have endocrine, paracrine, autocrine and synaptic. Now, endocrine means, first of all, let me tell autocrine. Autocrine is self-signaling. So, this cell only releases the signal, and the signal comes and binds with its own molecule, within the own cell. So, this is autocrine, self-signaling. Paracrine means it is giving signal to some nearby cell. Okay, but it is not far away, some nearby cell only it will be able to, the signals that it is released will be diffusing to that nearby cell and it will bind to the receptor. Endocrine signaling is a little bit more distance, that means the signal source will be somewhere and the receptor effect will be somewhere, means the effector cell will be somewhere. So they have to travel a certain distance through the circulatory system, and then that is how they can have that effect. Whereas synaptic is about the muscle and neuronal cells, where the signal transduction for nerve impulses will be sent. As you can see, these are the different types. So we have the contact dependent, which is short range, example in a nerve cell production. Then we have the paracrine, which is the local signaling, example for if any inflammation is initiated. So as you can see, this is a signaling cell which may have got damaged in case of some injury. Now this cell is going to release some signals, because whenever there is any damage or cut, there is a chance of bacterial infection. So the cells might release a warning sign for the other cells to be prepared. So that will lead to inflammation and recruiting of the immune cells to that site. So some local mediator will be released from the signaling cells, which the target cells receptors will take, and they will have the effect. Okay. Contact dependent, as I said, juxtacrine signaling, notch and delta. This particular signaling has a effect in development. Okay, it is important from developmental point of view. Endocrine receptor, these are long distance, example what we see in hormones. The site of secretion of the cell is a different site, and through the bloodstream it will travel, and the target cell will be again a different site. And the last one is synaptic, where we have this neurotransmitters getting released at the synapse, that is from the neuron to the target cell. So these are postsynaptic targets, can be muscles or any other neuron also. This is again a category of cell-to-cell signaling. Similarly, you have many ligands also, signaling ligands that bind to cell surface. Receptor is one type. Example, neurotransmitters like norepinephrine, histidine, or hydrophilic molecules, charged polar ones. Peptide hormones, example insulin, which cannot cross the membrane. Then growth factors will be there, different type of growth factors. Generally, these growth factors will be taken by the receptor tyrosine kinase. Okay, and lipophilic signaling molecules, example prostaglandins, will also be there. Other type of ligand that is there, as I said, that won't be cell surface, but these will be intracellular receptors. Okay, so these are mainly lipid soluble hormones that diffuse across the plasma membrane and interact with receptors in the cytosol or nucleus. That is steroids, thyroxine, retinoic acid and nitric acid, which doesn't have any problem going through the membrane. So why do they need cell surface receptor? They can directly bind to the intracellular receptors. Coming to the three important classes of cell surface receptor, one of them is ion channel linked receptor, where these channels are having two different conformation. One is the closed conformation, another one is the open conformation. If there are some, and what will be affecting these channels, like how it will be regulated, it is by a certain molecule or a ligand. So as you can see, only when this ligand is coming and binding, there is a conformational change in these channels which get opened, and then only the ions can move to the other side. Okay, but if the ligand is not bonded over here, then the ions cannot flow. One very good example of this is the endoplasmic reticulum ligand gated channel for calcium ions. Only when the IP3 ligand comes and binds to this ligand gated channels, then only the channel will be open and calcium ions will be released from the endoplasmic reticulum into the cytosol. The next category of receptor is G protein linked receptor. So the reason why it is called as coupled receptor is because two components work together. One is the receptor, which is generally a 7-pass receptor. 7 pass means 7 times it goes through the membrane. It is one single polypeptide chain. It's also called a serpentine because of the structure that it takes as a snake. And this is going to be coupled with another protein. Together they will be giving the signaling effect. This another protein is called as the G protein. As such, this protein is inactive, but only when the ligand comes and binds with the receptor, then only this G protein becomes activated. I'll show you how. And if the target protein is not bonded with the G protein part, then it is inactive. But when G protein becomes active, it undergoes dissociation and one of its subunit goes and binds to the target protein, thus activating it. This is the second important class, and another important class is the enzyme linked receptor. So here, as I said, the RTKs, they would have some enzymatic domains in their receptor, and they would be having some catalytic domains also which are active. Only when the ligand comes and binds with them, then only these catalytic units will undergo dimerization. As you can see, these two subunits have joined together and they will be active at this particular point. Okay. So the different classes of general types of signal transducers, as I mentioned, gated ion channel, then we have the serpentine receptor, then we have the receptor protein. Apart from that, you also have this adhesion receptors, receptor with no intrinsic enzymatic activity, and also the steroid receptors. Okay. So as of today, let's look at some more details about the serpentine receptor and the receptor tyrosine kinase. First, G protein coupled receptor and their second messengers. So this is, there are many details which you have to know for this process, as it cannot be covered in one video. If you want to know more details, you can refer Biotechnica recorded classes or online live classes that we have. Some batches are coming for the upcoming CSIR exam. You can enroll in them, then you can get the recordings, access of the recordings of the live classes, and you can even attend live classes where you can ask doubts to the teachers. Right? So a lot of details are covered in that. Let me tell you the most important points. So here there are three extracellular loops which are created in the G protein, as you, in the receptor, as you can see. So first of all, there are 7 passes, transmembrane passes of this receptor, and three loops will form the ligand binding domain, and 3 loops towards the inside, they will be binding the cytosolic domain. They will be forming the cytosolic domain for binding to some other proteins. Okay. The amino terminal part will be present towards the extracellular side, whereas the carboxyl part will be present towards the cytosolic side. Okay. Now, this is what is the transmembrane receptor which can interact with G protein. Next is the G protein. So this G protein is the effector enzyme that activates, means the G protein is the mediator which will be activating the effector enzyme. Okay. So here G protein is consisting of three subunits, which is alpha, beta and gamma. But there is why it is called G protein is because a GTP is linked with this protein. Okay, so GTP, it can be GDP also, it switches between that. So whatever it is, GTP or GDP, it is associated with this alpha subunit of the G protein. Okay, and as you can see that the alpha subunit and the gamma subunit are embedded in the membrane. Notice that beta subunit is not embedded in the membrane. Okay, now whenever this activation will take place, a part of this G protein will dissociate and it will go and bind with this effector molecule. So effector enzyme is something which will generate an intracellular second messenger. As I showed you in the previous pathway, that in such kind of signaling molecule, an effector molecule will release another secondary messenger. It will generate another messenger molecule. So in this case, the common type that we have is adenylate cyclase, the effector molecule, and the secondary messenger which it will make is CAMP or cyclic AMP. This can go and affect other proteins as well. Okay. Now let's have a detailed look at what is the procedure over here. What are the signaling cascade? Now this G protein, which I said contains alpha, beta and gamma subunits, this is called as a trimeric G protein. Okay. Here, the exterior will bind to the hormone, whatever ligand is there. So this is the resting stage. Even the effector is inactive. But when the ligand is coming and binding over here, then it is inducing a conformational change in the receptor, right? So because of this, the receptor conformation changes, and now this G protein with the alpha part, as you can see, it is interacting with the receptor. All these minute details are very, very important from exam point of view. So make sure you make a note of them whenever you are studying this concept. Activated receptor will bind to the G alpha subunit. Okay. Now G alpha subunit is right now inactive because it has a GDP. But when the receptor will bind this G protein, it induces a conformational change in the G alpha subunit, because of which it cannot bind GDP anymore. So as you can see, GDP has come out over here, and now this place will be empty, where immediately a GTP will come and bind. Okay, so binding of GTP to G alpha, whenever this GTP is coming and binding, it will trigger the dissociation of G alpha from the receptor and from G beta gamma. So not only it will separate from the G beta gamma, but it will also get dissociated from the receptor. Why? Again, because there is a conformational change. Initially with this conformation, it was able to bind to the receptor and also to beta gamma, but because of this GTP binding, it will dissociate. But this is now capable of binding with the effector molecule. Okay. In the meantime, hormone will dissociate from the receptor and G alpha will bind with it and it will activate. Okay. But the problem is that, okay, once the active effector, it is activated, it is going to synthesize the second messenger. All that is fine. But this cannot be for long, because hormone has dissociated from here, and GTP also eventually will undergo hydrolysis. So whenever this GTP will undergo hydrolysis to form GDP, it is the G protein only which can hydrolyze its bound GTP. Okay, so they have inherent GTPase in them. That's why they are called as G proteins. Fine. So whenever they are dissociating and then they are getting associated with a GDP, their conformation will again change. So whatever they were bonded with the active receptor, they will now dissociate and rejoin once again with their beta gamma subunits. So again it will form a trimeric G protein ready to go for another signaling cycle. So if you look at a animation over here, binding of ligand activates receptor. As of now, there is no hormone bonded. That's why the G protein exists in the inactive form, the trimeric form, and even the target protein is inactivated. But when there is the hormone bonded over here, G alpha binds with this and the GTP is converted into GDP. So beta gamma is also separated, and when this GTP is bonded, this alpha subunit will go and bind with the activated target. So heterotrimeric G protein binds activated receptor. It is called heterotrimeric because 3 different subunits are present in the G protein. Activated receptor acts as GEF. So what is GEF? Guanine exchange factor. Okay, so because it is removing the GDP and it is introducing a GTP. So whenever this receptor is activated, this is the reason why GDP is removed and a GTP is introduced. Because when the receptor bonds ligands, it will be activated and it can act as a GEF. Activated components, they will regulate the downstream targets as I mentioned. And GTP hydrolysis inactivates G protein, subunits will reassociate, and that is when the signaling process will switch off. Since it's a signaling process, there will be a switching on process also, a switching off process also. You should be aware of the both. Okay. So there are different types of G protein. Ones, as we saw, trimeric G protein, they are called as G stimulatory. Why? Because they will stimulate a particular effector signal, which is adenylate cyclase. The examples of such hormones are glucagon, beta-adrenergic, parathyroid. For example, when there is glucose signal, glucose concentration is less, then we have the glucagon signaling. So glucose is less means the cell should break down the existing glycogen stores to release free glucose, and also the liver should undergo the process of gluconeogenesis. That means gluconeogenesis, the glycogen breakdown enzyme should be activated, Glycogen synthase enzyme should be deactivated, and gluconeogenesis enzyme should be newly synthesized. Okay, so all these processes are brought about by the glucagon signaling through the G protein signaling, especially the G stimulatory, which is going to stimulate the adenylate cyclase, and CAMP will be synthesized. But that's not only all. You also have G inhibitory as well, like somatostatin or alpha-2-adrenergic. In these signaling process, it is not always activation of adenylate cyclase. In these cases, adenylate cyclase will be inhibited. So even these are important. There is another thing which is called as GAQ. It activates another cycle, which is the phosphatidylinositol, where we have the stimulation of phospholipase C, which we see in many of the instances, releasing IP3 and DAG. Just now I was talking about IP3. So that is another process which G proteins can activate. And this is another one which we see in rhodopsin, stimulation of cGMP phosphodiesterase. For CSIR exam, all these processes you have to study individually. Hence I said, if you are attending the biotechnical classes, then each one of them will be dealt in detail, because all of them are important and questions come regularly from this topic in CSIR exam. Okay, apart from that, you also have small G protein. Okay, so they are not trimeric. So that example is Ras in insulin signaling. You also have some other type of G proteins that we see in nuclear transport, example RAN, or in cell cycle, which we see Rho. So these are also categories of G protein. Even in protein synthesis and vesicle trafficking, RAB, these are all forms of G protein. So what is the G stimulatory pathway? As I said, it will be activating the adenylate cyclase. Now you can see this is a receptor where the hormone has come unbound. Glucagon can also bind. Epinephrine is also another example. The occupied receptor causes replacement of the GDP bound to G stimulatory by GTP, thus activating the G stimulatory. Okay, so the alpha subunit moves to adenylate cyclase and activates it. Now what about adenylate cyclase? Further on, adenylate cyclase will synthesize CAMP. It is made from ATP only. And the reverse process, it will be done by phosphodiesterase. Okay, because the ATP creates an ester bond to create the cyclic AMP. If you have to break this CAMP, then we would be needing a phosphodiesterase. So as you can see, it's written over here, cyclic nucleotide phosphodiesterase. Now why is CAMP important? First of all, before dissociation, it will create the signaling transduction. So CAMP further goes on to activate another protein which is called as pKa. pKa has two regulatory subunits and two catalytic subunits. The regulatory subunits bind the catalytic subunits, because of which kinase activity is inactive in inactive pKa. Okay, but CAMP has regulatory site has some binding site for CAMP. So whenever CAMP come and bind to this regulatory site, the catalytic sites, they will get activated and the pKa molecule will be able to phosphorylate other proteins because it is acting as a kinase. So once pKa is active, it will phosphorylate other cellular proteins and it will cause the cellular response, which is for the epinephrine. Okay. That's why I said G proteins are acting as molecular switches. Whenever the G protein will be binding with the, whenever there will be a GDP bonded with it, then it will be inactive. Okay. And whenever the G protein will be bonded with GTP, then only it will be active. So as you can see, monomeric G proteins are activated by GEF proteins. In case of G protein coupled receptor, the activated receptor was acting as the activated protein, which promote the swap of GDP and GTP. So like GAPs, GEFs are controlled by signaling pathways independent of the G proteins they will target. Now once it is activated, once it is bounded with GTP, it is active. Now this GTP should be removed also. So these are controlled by GAPs. These are GTPase cycles which are present. Monomeric G protein are controlled by GAP protein. They promote GTP hydrolysis. GAPs are activated by signaling pathways that function independently from G proteins. Alright. What are the examples of physiological processes which are mediated by GPCRs and heterotrimeric G protein? Example is epinephrine, serotonin, light, as I mentioned about rhodopsin receptor. The effector molecules will be different. For example, in rhodopsin, it is cGMP phosphodiesterase. As we saw for epinephrine, as it was adenylate cyclase, here it will be cGMP phosphodiesterase. The different physiological responses are also mentioned. As I already mentioned that you will have to study all these processes individually. So G-protein coupled receptor also activates IP3 and calcium mediated signaling pathways. So till here, as we know, the activated G alpha subunit, instead of going to the adenylate cyclase, it is going to come and bind with something called as phospholipase C. Phospholipase C acts as an esterase, cleaving the phosphatidyl inositol triphosphate inositol PIP2, which is a phospholipid inside the membrane. Once this is cleaved, you will get this IP3 and DAG, which is still embedded in the membrane, but IP3 gets free and comes and binds to the endoplasmic reticulum calcium gated channels, which I was talking about. On binding of the IP3, this receptor is open, the ion receptor, and the calcium channels will flow out. Now this calcium as well as the diacylglycerol, both of them can activate another kinase which is called as protein kinase C. So this is a different one from the protein kinase A, which was activated by CAMP. This particular kinase, protein kinase C, it is activated by calcium and DAG. I know there are a lot of components which are involved over here, but that's why this topic is not only important, but it is actually very interesting also. It's not that difficult. It's just that if you really have curiosity in this, you will be able to retain all the information. Okay. Then another important one is Ras-MAP kinase pathway. I will just summarize this particular topic. Cascade of cytosolic kinases that play significant roles in signal transduction. Ras-MAP kinase pathway cascade is a cascade of enzymes resulting in activation of transcription factors. Okay. So whenever we have this receptor tyrosine kinase I was talking about, this is how the receptor tyrosine kinase phosphorylates itself upon binding of the signal, and in this case the phosphorylated receptor is bonded by some other proteins. So one of them is GRB, which is the adapter protein. Adapter word here is in the same reference that we use in our some technical items, right? So how we adapt, we will connect one thing to another. Like that only here adapter is connecting one molecule to another. This another molecule is RAS-GEF, which is initially inactive, but only which is initially inactive, but upon binding with GRB adapter protein, it is able to be activated, and that is when the RAS will act as a GEF and it will activate the RAS protein, and further there will be some transmission along the multiple pathways. Okay, so the steps that are involved in a generalized MAP kinase pathway is the activated RAS protein is going to activate MAP kinase. The reason 3 kinases are given is because there will be 3 steps of phosphorylation. So as you can see in the first one, after MAP kinase, it will make MAP kinase because already a phosphate group has been attached. In the second step, 2 phosphate groups will be attached, and then MAP kinase will be able to phosphorylate some target proteins over here. So you can have the change in directly in the proteins also, changing the metabolism, regulation, and you can also have phosphorylation of some regulatory proteins which will affect the gene expression also. Okay, so here is an animation. When hormone binds to the extracellular domain, the receptors will then aggregate together. So this is the growth factor. When it binds to the receptor tyrosine kinase, then they are going to dimerize. Now not only dimerize, but then they will phosphorylate its parts. When the receptors aggregate, the tyrosine kinase domain phosphorylate the C-terminal tyrosine residues. This phosphorylation produces binding sites for proteins with SH2 domains. These are some specific type of domains present. So GRB2 is one of the proteins which have SH2 domain. GRB2 with SOS, which are bound to it, will bind to the receptor complex, which will cause the activation of the SOS. Once SOS is activated, it is a guanyl nucleotide release protein. When this is activated, there are some Ras proteins which are present. It will activate the Ras protein, release the GDP and exchange it for the GTP. Ras, whenever is bound with GTP, then Ras will become active in that case. Once Ras is activated, it will cause the activation of another kinase which is called as MAP kinase, also called as RAF1. RAF1 will further activate another kinase which is MAP kinase. Two kinases will be there. So that is also called as MEK. Okay, this is another cellular kinase. Now this further will activate another MAP kinase, activated MEK then phosphorylates another protein kinase called MAP kinase, causing its activation. This series of phosphorylating activations is what is a kinase cascade, and that's why it results in amplification of the signal. One small signal can be having an amplified effect on the target proteins. And among the final targets of this kinase cascade are some transcription factor. For example, here it is called as, it is shown Jun and Phos. When these proteins undergo phosphorylation, they will become active and then they will be able to give the physiological response. In their case, binding to DNA. Some other signaling pathways, important ones which you should be aware of is JAK-STAT pathway, TGF-beta SMAT pathway, NF-kappa B pathway, and from developmental biology point of view, Notch delta signaling pathway, WNT signaling pathway, and sonic hedgehog pathway. You should be aware of their functioning, the process involved, the elements, components, and also what is the physiological response. That's all for today. Let me know in the comment section if this has helped you in your CSIR NET Life Science preparation. Thank you.